Good afternoon. My name is Thea Guidry, and I'm the lab manager of GeneLab, which is located at the LSU School of Veterinary Medicine. GeneLab provides a variety of molecular biology functions, including DNA sequencing and real-time PCR functions. And we are an integral part of the LBREN Molecular and Cell Biology Core. One of the missions of GeneLab is to bring in speakers and scientists to share with Louisiana researchers various molecular and analytical technologies. In keeping with that mission, we have invited Mark Lawson, field application scientist for Bio-Rad Labs, for session 2 of our GeneLab molecular training session series. And he's going to present information on gene expression and real-time PCR. He will give real-world examples for analysis, troubleshooting, and optimization workflows using the new publication standards, the MIQE guidelines, which is minimum information for the publication of quantitative PCR experiments. And I turn the floor over to Mark Lawson.
Mark Lawson to talk about MIQE.
Yes,
I'm going to be jumping around quite a bit just to give the camera a hard time following me. We're going to be talking about quite a bit today, and we've actually got 2 sessions that we're going to be running back to back. I'm assuming I'm talking to the right camera here.
You are.
All of this technology is wonderful. As someone who spends an enormous amount of time traveling, the idea of coming here and being able to see all of you is a big advantage. So I'm glad that we could have this time to talk and that I'm able to reach so many different people from so many different areas. So today we're going to be talking about really from the beginning to the very end of the workflow for real-time PCR experimentation. And I'm particularly going to be focused on gene expression, that is fold change in various different conditions. So I have to start in any discussion with the very basics. And I know for some of you this will be quite a review, but real-time PCR, of all the different techniques or technologies that have impacted molecular biology, has been one of the most impactful in the last few years. A field that started in 1999, if you can imagine that, it's already gotten into almost every area of research, everything from botany to insect biology to studies of the brain to cancer diagnostics and as well as viral diagnostics. The particular applications all tend to focus around one particular strength of the technology, and that is its ability to quantify, its ability to answer how much questions, the kind of questions we might be asking, how much, if any, of the target sequence is present in the sample. We might also be asking quantitatively, how do treated samples compare to controls for a variety of different genes in different conditions. Some of you who've been doing this for a while or have been in molecular biology for over a decade will recall these assays. We once called these quantitative PCR, and we called them qPCR, in fact. And what we would do is take PCR, stop the reactions at cycle 20, 25, or 30, run the products out on an agarose gel, and do a simple densitometry analysis and comparison between the different samples. And that works. And that is quantitative, but it's only quantitative across a very narrow range. The real strength of real-time quantitative PCR, and notice I'm throwing in the word real-time now, is that it's very resistant to a broad range of different inputs. So we can have as little as 1 copy detected on the same plate that we're detecting 10 to the 7th or 10 to the 6th copies. And as you can imagine, there are some applications where this is a big advantage over previous assays that may have only had a dynamic range of 2 or 3 log orders. So it's really been very powerful for things like virology and cancer diagnostics. The basics of PCR, and again, I apologize for the deep review. If you want to think of PCR as essentially a Xerox machine, if I can use the brand name, a mimeograph machine or a copy machine, what we're doing is we're putting in a certain number of copies in the tube at our initial state. We're pushing the button, and each time we push the button, we get twice as many copies as we started with, in theory. So we have, let's say, initially a single copy shown here. We have all of the raw materials. So we have primers, we have DNTPs, we have a thermostable DNA polymerase. That polymerase chain reaction begins when we denature the template DNA to single-stranded DNA, which gives the primers access to their complement sites. So those primers are going to anneal to their complements as designed in the primer sequence. They're going to anneal to their complement sequence, and that 3' end provides the substrate for new DNA synthesis. So the polymerase, which is a DNA-dependent DNA polymerase, in most cases we're talking about thermus aquaticus or Taq polymerase, extends that primer molecule to make a short copy of a particular sequence between the forward and reverse primers. The net effect is that for every 1 copy we put in, every cycle, we theoretically get a doubling in copy number. So you can see at cycle 1, at the end of that cycle, we went from 1 copy to 2 copies. At the end of cycle 2, we go to 4 copies. And at the end of cycle 3, we go to 8 copies. And that number, that expansion continues almost indefinitely until we have a very large number of copies. So again, if we started with 1 copy, we end up with 2, and then 4, and then 8, and 16, and 32, and 64, and 128, and so on, so that by the time we hit cycle 15, we have 32,000 times as much as we started with. By the time we hit cycle 20, we are at 1,000,000 times the number of copies we initially had. And you can imagine filling in, for example, 1,000, we would now have 1,000,000,000 copies. By the time we hit cycle 30, we have 1,000,000,000 times as much as we started with, and it goes on and on. If we continued indefinitely, we could theoretically have, I think by cycle 200, a crystal of DNA that was larger than the size of the Earth. It's the power of exponential growth. Of course, we're not going to do that. And the reason we're not going to do that is because we don't have enough raw materials to make a crystal of DNA the size of the Earth. But the expansion here is highly predictable from the nature of how PCR works. So that gives us what's called the first principle equation, which is the initial quantity, that's Q sub 0, times 2 raised to the number of cycles is equal to the quantity at any given cycle. So that's essentially what this graph is, or what this list here is illustrating. Using that equation, we can back calculate how many copies we started with. So we're essentially solving for Q sub 0. With that in mind, oops, I've gone too far. I've given away the answer. With that in mind, we can already begin to see how PCR-based quantification can work. If I have 2 samples that differ, where the first sample is at 15, I've discovered that my hand is also a pen, at cycle 15 for sample 1 and cycle 18 for sample 2. Those 2 differ at a particular point, right, where they've achieved the same copy number. They differ by 3 cycles. So we would express this as the delta CT value, and I'll talk about that in a minute, of 3 cycles. Anytime we want to convert from cycle numbers to fold change, we use a power of 2. So in this case, we have a difference of 3 cycles. That's 3 rounds of doubling, right? 3 times pushing the Xerox button. So that's 2, 4, 8. It's perfectly acceptable to do it on your fingers, right? Go back to kindergarten math. 2, 4, 8. So I would say the difference here is an 8-fold difference. Well, I can of course skip to the next step, but I have to finish my word. A delta of 3 cycles is an 8-fold difference. All right, and that's the kind of comparisons that we're going to end up doing in our gene expression experiment. A direct comparison of 2 samples. But we can take this a little further. Let's suppose that we have our no template control. This is a sample that doesn't contain any cDNA from our samples of interest. Instead, we've used water. So this is our blank control, or our no template control, and it's positive. And this is something that quite often happens. But our lowest sample is at cycle 29. It's detectable at cycle 29. Right? Our no template control is detectable at cycle 36. With qualitative PCR, with conventional PCR, that would simply be viewed as a failure, right, a problem. With quantitative, with the ability to quantify these amounts, I can actually say that this is a delta CT of 7 cycles. And since I know my powers of 2 math, I can say 2, 4, 8, 16, 32, 64, 128. So I can say that the difference between my no template control and my lowest abundance sample is a 128-fold difference. And I can express that in my manuscript, my grant submission, wherever I'm documenting this, as less than 1% of the signal in the lowest sample could have been contributed by non-specific sources. So rather than arbitrarily saying that I don't worry about false positives so long as they go past cycle 35, I can actually quantify how wrong or how much of a contribution that signal is to my total signal. And we'll repeat this process when we do something called the RT minus control. How about a paper that we're reviewing where you can visually see that within a replicate we have a variation of half a cycle? What fold change does that represent? Right, before we were working with whole numbers. We had easy math, right? We could do it on our fingers. Now, though, we want to express what is half a cycle in terms of fold change, right? And we're going to use the same kind of thing, delta CT of 0.5, and we're going to express that as 2 raised to the 0.5. And who knows what 2 raised to the 0.5 is? Anybody?
1.
Up a little more. That's the square root of 2. For those of you that weren't educated in the Texas public school system, you might know that number.
1.4.
1.4. 1.4, and that's good enough for a biologist. 1.4. I don't want too much math. So if they're saying that they see a 20% increase in copy number, and the plus or minus range here is 1.41-fold or 4-fold, then you know that they're going to have overlapping error bars. And that's a simple way to check that things match up with your expectations. All right, so we can use our powers of 2 math to convert cycles into fold change. This is what the curves actually look like. Getting a bit of an echo. This is what the curves actually look like. On the y-axis, we have relative fluorescent units. That is relative fluorescent units. That means that the actual intensity of these values is irrelevant. It's there primarily to give shape to the curves. On the x-axis, we have cycles. So cycle 2, cycle 4, cycle 6, cycle 8, and cycle 10, every 2. All right? Just like a bacterial growth curve, this curve has 3 portions to it. We have the pre-detection phase, which I would say covers about to cycle 14 here. In every round, we're still doubling, but we're not able to directly detect it. We then, though, the slope begins to change, and we enter the linear or exponential phase. And I call it the linear phase because it has a constant slope, and that slope is actually something we want to keep an eye on because it tells us in a ballpark way what the efficiency of our assay is. Then we enter a plateau phase. If we never entered a plateau phase, we could, of course, we have that Earth-sized DNA crystal. Now, why does the reaction begin to plateau?
Using up parts of your reaction.
Exactly. We're depleting our resources. There's not enough nucleotide. There's not enough primer. There's not enough Taq polymerase to make an infinite amount of DNA. So at some point, we're going to run out, right? And that's when our slope is going to begin to change. And I've drawn it up here, but in fact, the slope has begun to change as early as here. And you see your replicates, and this is a replicate of 4, begin to diverge. So by the time we're very midway through the linear phase, the slopes have already begun to change. So if we measure at this point we're always going to have some variation inherent in depletion of resources. And that's not what we're interested in. We're only interested in when everything else is in excess except the template. And that's why we're going to draw a threshold line. That threshold passes through the earliest part of the linear phase. Right? If you want to imagine this as an arm, this is the first part that is on your upper arm that's not part of your elbow. It's the first place that the slope has begun to flatten out. And we actually do this in our software using calculus. We take a partial second and partial third derivative analysis of this graph. The maximum on the third derivative is, the maximum on the second derivative is the point of maximum inflection, and the minimum on the third derivative is the first point on the curve that is completely flat. That, in other words, it's in a trajectory. And we pick a point somewhere in between. I've done a lot of drawing here. Uh-oh. There it goes. So we've chosen a threshold based on the shape of the curve, not on the fluorescence value. But it does, in fact, have a specific fluorescence value here. And wherever the samples intersect that threshold line, we're going to have a CT value. Or the new term is, forgive my handwriting here, is a CQ value. And that is the quantification cycle. The cycle at which we cross the threshold or the quantification baseline. All right? Part of what I'm going to be talking about today is MIQE. This is the new publication standard. Part of the MIQE standards means changing our terminology to something that is not company specific. And CQ was deemed to be neutral enough to be usable in any publication. So prefer that you call it CQ. Those of us that have been doing this for a long time have a hard time changing over. So you have to catch me when I say CT. Now it's interesting, but not all that important, that you note that the CT value for this is 15.8 for this replicate group. That is, we measured it and we determined that the intercept point is at 15.8. But there's not really any such thing as 15.8. 15.8 doesn't really exist. Right? Can anyone figure out why there's no such thing as 15.8?
Because cycles are discrete.
What's that?
Because cycles are discrete.
That's correct. Very good and very well said. So the cycles themselves are discrete. There's no such thing as 0.8 of a PCR cycle. And in fact, we only measured it at 15 and at 16. We didn't measure it in between cycles, whatever that would mean. So in fact, we measured it at 15, we measured it at 16, and sometime during that cycle, we would say it crossed. But of course, again, it's not a continuous process. So what we've done here, and we don't do this on purpose, but we've drawn a smooth continuous line that's actually made up of 40 points. And we've drawn a line in between those points. So what you're really measuring at 15.8 is an interpolation between two measured values. And that means that the smallest difference between any two samples that we could directly detect is one full cycle. We've been talking about powers of 2 math. What fold change is represented by a one cycle shift? 2, right? So the smallest directly detectable difference using real-time PCR is a 2-fold change. Anything smaller than that requires interpolation. And this is called the 2-fold limit of resolution, right? We still use the data, but it has to be based on accurate interpolation of points on a curve. All right, so real-time PCR is not all that great at detecting very small differences, less than 2-fold changes. It's fantastic at the larger amounts, but not so great at the little ones. Now, we can also take this curve and log transform it. And this is more often how you'll see it in the literature. All we've done really is change the value on the y-axis to be log transformed. But otherwise, the data is the same. Of course, we compress values down here. and we expand values up at the upper range. So, 10 is obviously somewhere around here. Okay? So that distorts the shape of the curve. My own recommendation is use the linear curves to make sure that your thresholds are passing through the right shape, that is just above the elbow of the data, and then flip it over to amplification. Now, what good are CT values? We know how they're obtained, we know what they mean, but how do we use them? The clearest illustration of why we use CQ values is illustrated by a standard curve. And if you get nothing else from the presentation, I hope you get the value of a standard curve. So what I've done here is I've got a 10 to the 8th copies of a plasmid in a tube. That's my first sample. And then I diluted it 1 to 100. And I diluted it 1 to 100 and 1 to 100. So I've got 10 to the 8th down to 100 copies on the same plate. And that's a pretty broad range. And so each of these is 100-fold apart at their initial copy number. What does the spacing look like? Is it even or uneven? Does it look pretty even or pretty uneven? It's very even across an incredibly broad range. And in fact, we should be able to predict how far apart these CT values should be if we're truly doubling in every cycle. And the equation would simply be 2 raised to the what overcomes the 100-fold initial difference. And who can do that in their head? I do it on my fingers. 2, 4, 8, 16, 32, 64, 128. So somewhere between 6 and 7. So in fact, it's 6.6. These should be 6.6 cycles apart if every cycle is truly a doubling. And in fact, we're at 10. We're at 12, 14, 16 and a bit. 16 and a bit out to about 23. And then 23 out to not quite 30. So we are pretty doggone close to that theoretical doubling. If these are a little further apart, then we know that the actual copying efficiency isn't 100%. It's less than 100%. If they're closer together than 6.6 cycles, then we're getting an efficiency of copying that is higher than a doubling, more than 100%. And knowing that number will improve the quality of your data, both when you're using standard curves and when you're not using standard curves. When you're using a 2 to the minus delta delta CT analysis, which most people are doing. Knowing the efficiency prevents you from making assumptions that are false. And that's really what standard curves are especially good at, is testing our assumptions. Let's take the observed CT values and the given concentrations or quantities or copy numbers, and we'll graph them against each other, but we're going to take the log of the quantities. This gives us a standard curve, right? This is our normal looking standard curve plots. Quite pretty, actually. On the y-axis, we have the CT value. I didn't actually mean to cross that out. On the x-axis, we have, and this is very important, the log of starting quantity. So we're log transforming, log regraphing in a log. So 2 here now represents 100 copies. 8 represents 10 to the 8th copies. 100 copies is not a lot of DNA. 10 to the 8th copies is a lot of DNA. What shape is the relationship between CT value and the log of starting quantity? The sun, a moon, a clover, heart. It's a straight line. And in fact, we can measure how good a fit is the data for this straight line. And we do that with the R-squared value, which is given here in the graph as the coefficient of determination. We need an R-squared of greater than 0.95 for this data to be usable. If you don't, then you're using a model. You're making assumptions that are not true. And we need to know that the R-squared includes the range of CT values we're getting for our samples. So if this is my assay, And I have validated it now from cycle 30 to cycle 10. If I suddenly get a CT value of 32, I cannot assume that this line continues on forever. And that's when I would have to reject values of 32 because I have not validated the assumptions, not just for standard curve analysis, but also for our relative quantities. So this is the value of a standard curve. It's testing whether or not our assumptions are true. The other thing we need to get is the efficiency. The efficiency is the actual copying efficiency. So it needs to be between 90 and 110%. Less than 90%. Who can think of some reason why it might be less than 90%? I can see you guys easily. What about bad primer design? Is that what you're going to come up with?
I was going to say something with the primers I couldn't word it.
Bad reaction conditions, not enough materials, the presence of an inhibitor in the samples. Almost anything that you've got suboptimal will result in copying efficiencies of less than 100%. So we need to know what those things are. And if we're getting less than 90% efficiency, you're going to have large standard deviations and big error bars in your analysis. So we want to keep it greater than 90%. But the other one, and maybe a little bit more common, in fact, is less than 110% efficiency. And every once in a while, we get a phone call. People want to know if they've hit 200% efficiency, if they should call the Nobel Committee or wait for the Nobel Committee to call them. We have to break the bad news to them. 200% efficiency is just as bad, if not worse, than, say, 30% efficiency. Right? Now, some of you that have done real-time probably know what the most common cause of ultra-high efficiencies are. Anybody? Primer dimer. Number one, any non-specific product being simultaneously amplified means that not only are we making a doubling of a copy of the product we wanted to make, but also other products as well. And the combined efficiency is greater than 200%. All right, and that, of course, is useless to us if we're trying to analyze a specific gene and we're getting multiple products being made. Okay, so greater than 110% usually has to do with non-specific products being made. Now, we've talked about that. We're going to keep coming back to it, but I want to go ahead and talk about MIQE before we get too deeply into other topics. For those of you who have not been following this particular topic, MIQE stands for Minimum Information for Publication of Quantitative Real-Time PCR Experiments. Now, I think they've skipped a few too many letters. I think it would actually be MIQE. Maybe that's why they skipped it. And I can't decide if they're making a reference to the Life cereal commercials with the Mikey hates everything in the cereal, and they hand the kid the cereal and he really likes it. Because I don't think a lot of people are going to be all that thrilled about MIQE.
No.
This is the publication. It's not a major journal, Clinical Chemistry, 2009. But the names on here are major. For some of you that are specialists in your own particular field, I'm only a specialist in real-time PCR, and these are all the rock stars of real-time. So people like Tanya Nolan, who has a very highly cited paper, Stephen Buston, who writes all the review articles on real-time, Jovan Ensemble, he has the 7th most viewed paper in Medline. He started a biotech or a bioinformatics company that does nothing but real-time data analysis programs. So Michael Fathel, I think, runs the German Gene Quantification Center. These are all the giants in real time, and they've all signed their names to this document. I should mention that if you are writing a manuscript right now for Nature, or for any of the Nature journals, you will notice this has been added to the instructions for authors. So already has been implemented. And we've had a number of other journals announce that they will also adopt these new publication standards. All right, so what is entailed in publishing this data? there's a checklist that you have to fill out when you submit your manuscript. Part of that will also be submitting your raw data, and that's a bit of a challenge for some people. I'll highlight what are the most important changes, the ones you probably are going to have a hard time implementing without a little bit of a rethinking of how you're doing your research now. All right, this is the easy stuff. Experimental design. You define your experimental and control groups, number within each group. Assays carried out where and by who. Acknowledgement of each author's contributions. I think there was some talk about free writer authors on key papers. The description of the samples, how they were processed, how long they sat in the freezer. How long they sat in the freezer is the most important one. Right? If we're comparing RNA that's been in the freezer for 6 months to RNA that's been in the freezer for 6 years, we're going to get very different results. Right? RNA is very labile. It breaks down quite quickly. Even DNA, over extremely long periods of time, begins to break down. And that affects what we assess of the relative fold changes. So it's very important that we know this information. And of course, one of the hottest areas right now is people taking pathology blocks or formalin-fixed paraffin-embedded samples. And these tend to be of very low quality. And we need to be able to, at a glance, see that they're using that kind of material.
Mark, what is the E and the D in that column?
Oh, I'm sorry. Thank you for asking. E stands for essential, D stands for desired. So essential is has to be done, and desired is optional but highly recommended. Thanks. I would have gone right past it. So next area, and that wasn't the whole form, by the way. The next area is nucleic acid extraction. So how are you extracting your RNA or your DNA? Did you DNA or RNA treat your samples at any point? Have you checked for the presence of contamination? And that might be as simple as trying to amplify a non-specific gene, running a no-template control. Then you have nucleic acid quantification. This is spectrophotometric analysis. So that means the nanodrop, which everyone loves. But that's not enough. This is one of the areas that most people will have a little trouble with. And that is, we need a whole separate section for RNA integrity. Remember that spectrophotometry is not adequate to tell us if the RNA is still intact or completely degraded. The absorption, the spectral absorption of nucleotides is irrespective of whether or not they're still part of an intact chain or polymer. Okay? So we can't simply take the 260, 280 and say that the quality of our samples is good and move on past it. Now the best technology for this is either an adjuvant bioanalyzer or, company plug, Bio-Rad Experion. Sorry, I have to make some money somehow, right? But those are the two instruments that are recommended specifically in the MIQE guidelines. But we'll talk a little bit about how those work, because I think it's an important technology to understand. As of today, you might be familiar with the Miami standards, especially if you're working in microarrays. And one of the requirements to publish any microarray data is to have this kind of technology enabled, so that you analyze your RNA for its intactness. The other thing here is inhibition testing, and that is as simple as running a standard curve. We'll talk a little more about that in a minute. Evidence of optimization. Now this is only desired, but even if you are buying a commercial assay from a company and they tell you that they've optimized it, they still would like to see the evidence that it has been optimized. And part of that is other people are going to be using your work to build on. So we'd kind of like you to go ahead and verify that is the optimal conditions to run your assay at. Okay, because it's going to be added to the body of scientific literature. The specificity. So that's as simple as running a gel. And I'll show you guys in a little bit a two-plate optimization scheme that includes a simple gel analysis of your products, which satisfies the requirement for specificity confirmation. For Cyber Green, you must have a no template control, and they very clearly specify on every plate. So if you're not currently running negative controls or no template controls on every plate, you should start. Standard curves with slope and y-intercept. Okay, this is all satisfied with a standard curve. PCR efficiency calculated from the slope. Confidence interval, R squared of standard curve. I just showed you a standard curve. Doesn't mean you have to run it on every plate. You only have to run it once for each new assay. So it's not a real cost or a cost in money or time. The linear dynamic range, which also is obtained, and actually the limit of detection as well, is all obtained from a standard curve. So every assay you ever run needs to have a standard curve conformation. And you need to gather this data from that standard curve analysis. Okay? Also on here is multiplexing. If you are multiplexing, and that's fairly rare in today's market, efficiency and limit of detection for each assay needs to be specified. Next up is data analysis. How are you analyzing your data? What program do you use? What are the results of your no template controls? This seems a little redundant to me. This is the one though, right here, big star. Justification of number and choice of reference genes. 90% of people right now are using beta-actin, GAPDH, or 18S, and they're using one reference gene, and they're using it because
It's historically what people have used.
because it's historical, right? It's traditional, or I picked it out of a hat, or I'm referencing another paper, and that's what they use. All right, and there's been a daisy chain of people just arbitrarily choosing housekeeping genes. And there really is no such thing as an invariant gene, right? Every gene responds to something. Somewhere there's a promoter, and that promoter has some binding factor that makes it effective, right? That recruits a polymerase. So everything responds to something. And we need to make sure that if we're going to normalize our data, that we're normalizing it to the right kind of gene or the right specific gene. And we might want to think about using more than one. Genes go up and down, they respond. So choosing a single gene means we're at the whim of that gene's response. If we're using multiple genes, they tend to cancel each other out until you get kind of a consensus level of expression. And so multiple reference genes is the current trend in gene expression. Number and concordance of biological replicates. There's quite a bit of discussion on whether or not 3 biological replicates is even worth doing. Number and stage of technical replicates. We'll talk about this in a minute. Repeatability, reproducibility, power analysis, statistical method for result significance, software, and then this is a fun one. And this is only desired right now, but they've announced that they're going to make this an expected factor, and that is your raw data submitted along with the manuscript. So that means the referee, the reviewer, can flip through your data in a raw way and see which wells you dropped out, which ones were excluded, right? For some people, this is going to raise a lot of questions, right? A lot of questions going back to the author about why would you drop 1/3 of this plate out, or why are they arranged in this particular way, or did you run an inter-run calibrator, or I don't see a no template control in this plate. All right, so for some people, that's going to be a fairly labor-intensive change to how they're currently doing research. RDML stands for real-time data markup language, and it's the equivalent of HTML or XML for real-time data. All right, and there's an RDML project that you can go to. It's rdml.org, and they have a place where you can upload Excel files containing your raw data. And we'll convert it to an RDML format. And that can go out to the reviewers. Any questions about MIQE? It's a lot to take in. And as I say, you're already being affected by it, those of you that are preparing your next Nature publication. Okay? There are 2 chemistries. We have cybergreen or DNA staining dyes, and then we sort of have everything else. And I want to talk about them one by one. So we're going to start with cybergreen and the DNA staining dye. Cybergreen was the original. Of course, before that actually was ethidium bromide. And people did actually use ethidium bromide in real time long, long ago. Cybergreen has about 5 times the sensitivity. There are newer dyes that are 5 times as sensitive as cybergreen. So it's one of the things we'll be talking about, evergreen, as part of some of the products Bio-Rad is putting together. The way it works is pretty common. You hit them with an excitation energy in the presence of single-stranded DNA or free in solution. When you hit them with that excitation energy, they re-release it mechanically. So they essentially rotate, they spin. If they're in the presence of double-stranded DNA, then they have a tendency to intercalate into the double-stranded DNA. Now they're locked. They can't spin anymore. They're locked. They're jammed in there. So now when we hit them with excitation energy, they have to re-release those photons at a slightly different wavelength. And that's what the instrument is detecting, the presence of that second wavelength of emission. But you'll notice that it's not specific on sequence. It's just the shape of DNA. So whether it's the product you wanted to make or some other product that you didn't intend to make, they both contribute equally to the fluorescent signature in the well. And so that means that this is a very non-specific technology. It is, however, still 90% of all the reactions run in the US, according to the surveys we've seen. Why is that?
It's cheaper.
It's cheap. It's inexpensive, I think is the appropriate term. It's inexpensive. It doesn't add a large amount of cost. Whereas everything other than the DNA staining dyes is going to cause a substantial increase in the cost of each reaction. So the mere fact that we all have limited grant budgets means that we try to get the most amount of data out of our reagents. The CyberGrain is the most cost effective. But it has a lot of downsides. It's not specific. It's not multiplexable, right? One other advantage it does have, what was that?
I'm just saying, right, it's not multiplexable. Sorry.
The other advantage it has is the ability to do melt curves. And this is my favorite feature. Right? As someone who has poured more than a few hundred agarose gels in my life, this takes the place of routine gel analysis of our products. We're going to find out their purity, their rough size, and we're going to determine how many products are being made in the tube. So the process is, at the end of PCR, we've completed PCR complete. We've got as many copies as we can possibly make. We're going to heat the samples very slowly from 55 out to 95 degrees. So the temperature is increasing. All right, it's going up. The fluorescence, as you can see, has a constant negative slope for the first 80 degrees here. But then it suddenly drops. That's the interesting part. And then, of course, we reach 0 and it stops. But this constant negative slope is distracting. And so we're going to use our good friend calculus to make this a little bit more friendly to look at. Now, the reason why the slope is continuously dropping has nothing to do with the DNA. It has to do with the buffer. So the buffer, as it gets hotter, becomes more acidic, right? Buffers that are hot buffer less. And that's why we're getting a constant decrease here. What we're really interested in, though, is where double-stranded DNA goes to single-stranded DNA. That is the sudden drop, right? The sudden decrease in fluorescence. We're taking advantage of CyberGreen's DNA staining properties. So here is the same graph, but this time we've taken the derivative of the previous graph, again calculus, and we flipped the sign. So that constant negative slope is now a slightly positive line. That sudden drop in fluorescence is now a positive peak. So it's very easy to say something melted here at about 83 degrees. And that's distinctive for this product. Now here's a quiz question. I love quiz questions. Can anyone name some things that determine the melting temperature of a particular DNA strand, or DNA molecule?
GC ratio.
The GC content, very good. What else?
Size of the
The
product.
size of the product, very good. The size and the GC content are the two primary determinants of the melting temperature of the DNA. There's some other ones. There's the ionic strength of the solution, which usually is constant. And there's also interactions between the molecules. And we can actually use that to our benefit in other applications. Primarily, though, the longer the product, the higher the temperature it melts at. And that's primarily what we're looking at here. But I can't calibrate this to molecular weight. So all I'm looking at here is just a giant check. Good product. And I know it's a good product because I have a single high temperature melt peak. Those are the two factors I'm looking for, single peak and high temp. What's high temp? Here's bad data. All right, this is actually two assays on one plate. So this is product 1 and this is product 2, and you can see they don't actually overlap for any of the samples. This, on the other hand, is a low temperature product. All right, and I'm going to give you an arbitrary number to use, about 80 degrees. In fact, a lot of products actually will tend to melt around 78 degrees. So it's not a perfect guideline, but I'm suspicious of anything that melts below 80 degrees. Right, I might drag my gel rig back out, pour myself an agarose gel, and verify the molecular weight of the product. Anything greater than 80 degrees is most likely a templated product. It may still not be the right product. You might be amplifying a pseudogene, which usually makes for some interesting comparisons. But it's still a templated product. So as long as I get a single high temp peak for every amplicon, I'm okay. But again, it doesn't have a lot of information to it. It won't tell us what the size of the products are, and it won't tell us specifically what the molecular weight is. So it's very, very great, very good to have when everything looks great. It's not so wonderful when you don't know what's going wrong. Okay? So that's my favorite feature of Cyber Green. There's everything else other than Cyber Green. And they're differentiated in that they're usually more specific. Not always. The generic probe chemistry is TACMan. That's the one most people are familiar with. And that's a particular brand name for a dual-labeled hydrolysis probe. Right? There are a number of other ones. There's displaceable probes, and then there's probe incorporated directly into the primers. Remember here, including this, we have 3 oligos. Here we still only have 2 oligos, 2 oligonucleotides. Right? The advantage of the 3 oligo system is that all 3 events have to occur. All 3 oligos have to find their complement for a signal to be generated. So it's more specific. It's also more expensive. So what does FRET stand for? I used to make this a quiz question. FRET is the interaction of 2 dye molecules. Most people will tell you that it is fluorescent resonance energy transfer if they're familiar with it. But in fact, poor guy, we have stolen this guy's name from history. His name is Theodor Forster, if I'm saying the German correctly. And he discovered that when 2 dyes were close enough together, that they're on the atomic scale close together, when we excite one dye, the energy is transferred through, and I'm going to pretend I know what I'm talking about here, through a dipole-dipole coupled mechanism that is non-fluorescent transfer of energy from dye 1 to dye 2, so long as they are very, very close together. And that distance, of course, is measured in Forster distance. So we have a probe in our PCR reaction, if we're going to use a TACMan or other probe chemistry, and it has 2 dyes that are within a FRET distance of each other, that are close enough together that this donor dye, which is bright, and the acceptor or quencher dye interact with each other. I'm exciting this dye, but I'm getting a signal off the acceptor dye, and I'm not looking for that signal. So we're calling that quenched, when in fact we're just shifting the wavelength out quite a bit to the red. All right, so I've got my interesting looking probe here. And I've also got my forward and reverse primer. So there's 3 oligonucleotides in this tube. What looks like Superman here is actually my Taq polymerase. And it does have to be Taq. And the red cape he's got on is the inactivation antibody. So this is an antibody that blocks the active site of the polymerase. We're always using a hot start Taq polymerase here. So I'm going to heat the sample up to 95 degrees. So you can see we've denatured the DNA and we've made the capes go away, and now our polymerase is active. You do not need to prepare your reactions on ice. We're always using a hot start enzyme. And in fact, preparing on ice often contaminates the bottom of the plates. So I highly recommend room temperature setup. Okay, we're up at 95. We've got the probe, and we've got two primers, forward and reverse. Now, the TM of the primers, let's say, is 60. Then the TM of the probe needs to be, I think, 66 to 70. I'm going to say 68 to 70. So it's substantially higher binding affinity. That's because the order of operations here is critical. And this is where most people mess up probe design. So at around 70 degrees, the probe is going to anneal between the forward and reverse primer sites somewhere on the complement or on the target. Now as the temperature continues to slide from 95 to 60, the forward and reverse primers also anneal. So the stage is now set. And I have to warn you, there's a joke coming. It's not a very funny joke, but it is a joke. And that is that we have a 3' hydroxyl group here. And that's the substrate for new DNA synthesis. All right, what's going to happen when I start making new bases of DNA and that Taq polymerase bumps into the probe? Yes. And the only way you can answer that question is if you knew that the polymerase has a 3' to 5' Pol polymerase. And it has a 5' to 3' exo, or exonuclease. And here's the joke. They thought it looked a little bit like that yellow video game character. Do you guys?
What? Pac-Man?
Pac-Man.
Ah, that's the joke. That's the joke.
Chomp, 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 chomp.
Yeah, so someone has to supply the sound effects for me. There goes our Pac-Man. There goes chompa, chompa, chompa. Wonk, 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 wonk.
Oh, I can't do the after effects. Yeah, there you
Little
go.
ghosts.
All right, so I now no longer have die 1 and die 2 close enough together that they're going to quench each other. This is going to be a quenching reaction. So every time I make a copy of this gene, I'm releasing one more dye molecule into solution. So there should be a 1-to-1 correlation between copies and the number of dye molecules that I've released. Now what would happen if the TM of my probe, bringing back Pac-Man here, if the TM of my probe were, let's say, 62, right, and the TM of my primer is 60. Or we can do it even further. We could say, what if the primer goes on, find this complement site, before the probe does? Will we make a new copy of that target? Will the polymerase make a copy?
Yeah.
Yeah, definitely. So we'll make a copy, but the probe won't be there to be degraded. It won't be in the way of the train, right, of Pac-Man. And as a result, we'll have the same number of probes, but we'll have an increase in copy number that's undetected. So the net effect of that is undetected amplification. So if you are so good at designing PCR primers, your PCR primers will anneal at temperatures 8-9 degrees above what you've designed them for, then you can end up ruining your Taq-Man reaction. So we need primers that are fragile. Now, how could we possibly test whether our primers are annealed at 68 degrees? How would we figure that out?
That melting curve will give you some idea.
It's easier than that. What if we ran the reaction at 68 degrees?
I was thinking a gradient PCR.
Well, you could do that too, yeah. You could run a gradient. You could run, just basically test the reactions. If you make a product at 68 degrees, then you know that your primers are binding. And if that's happening, then these primers are incompatible with that probe. And this is something that people often have to go through, even with commercial assays. Right? Usually the company hasn't completely worked all the bugs out of the kits before they sell them. I want to diverge just a little bit and talk about instrumentation. Not ours, I mean, we will talk about ours, but just in a generic sense. What you want to look for when you choose the instrument you're going to run on. There have been, I think now, 4 generations of systems. We're in the 4th generation now. Bio-Rad has recently launched our 4th generation system. The components, though, we have a thermocycler, which we want to be fast, uniform, and to have the right capacity and format. We have an excitation source, which is usually either a lamp or an LED. We have a detection source, which is usually a CCD camera or a photodiode. And we want both of those to have a broad range, be specific, and have sensitivity. And then the analysis software. And that is, of course, your experience with the instrument, in most cases, is going to be limited to running the software and doing the analysis. So this is a different variety of the instruments that we have. We have a 48-well instrument that is LED-based. And this is actually one of our current generation systems. We resurrected it with some new software. This is our current mainstay instrument. It's the CFX, and we have it in 96 and 384-well format. And it's specifically designed to be capable of high-level multiplexing, designed from a hardware perspective with that in mind. It's also really, really fast. I don't know if you guys have been keeping up with some of the newer conditions, but now it takes about 30 minutes to run 40 cycles without any special reagents or special plastics or anything else. So things have really changed in the hardware department. And so also in the reagents, and I will actually talk about that. But in this case, we've got LED excitation and photodiode detection, and each of those is filtered on the way in and the way out, so they're actually paired. And then we have a slightly older system that we're still selling called the MIQE 2, and it's a two-color instrument that is based on CCD detector. It's based on excitation filters and emission filters, and we have a lamp excitation. And again, filtered on the way in and the way out. One important distinction, I say this is an important feature of your hardware, is there's been a break. So you're going to see most new instruments are going to be coming out designed for low-profile plastics. So if you are looking around or if you're borrowing someone else's instrument, make sure that you differentiate which of the two plastics it's compatible with. The one school is low-profile plates and the other school is full-profile plates. The difference here is that these can only hold 150 microliters, whereas these can hold 250. Now, none of you, I hope, are doing 250 microliter reactions or even 200. So you can see why we really don't need 250 anymore. And the disadvantage of these full-height plates is that we need a much larger block in order to accommodate their height. And the more metal we have to have in the block, the slower we can make it ramp, and the slower it reaches equilibrium. So that's what's holding the technology back right now. For a lot of platforms. I also want to talk very briefly about the Experion. I did make a reference to it. If you guys don't mind, I'm going to sit down for a second. And the Experion is particularly for the analysis of RNA, DNA, and protein. Go this way. And where it fits in the workflow, you would do your RNA isolation and purification, and then analyze the quality of that product using the Experion, and then decide to either go to microarray analysis or real-time or both, usually. The way it works, I'm sorry, I should back up one step. It is based on gel mobility, so the actual restriction of the sites so that they can be separated on size. Was there a question? Okay. On spectral absorption, we only see how many nucleotides are present and the 260-280 ratio, but that's not telling us the intactness of the RNA we're working with. And for that, you need either the Experion, the Agilent, or polyacrylamide gel electrophoresis. The way it works is we have a chip that has on it, I'm going to draw in some little things you actually can't see, and those are the little wells. And the samples are placed in the wells, and then the chip is placed in the machine. There's a certain amount of sample prep that goes into it. And so 1 microliter of RNA, for example, can be analyzed. And the way the data comes out is an electropherogram, which is then translated into a virtual gel. I'll show you a little bit more detail. Underneath the wells of the plate, showing here, this is where you might place your sample, are a series of channels etched into glass. There's a little glass slide cover underneath that has been etched. And all of these channels are filled with linear polyacrylamide matrix, which separates the products out based on their mobility. So we're going to electrophorese the samples through these little channels, and they're going to go to specific areas, for example, where they are stained, where they are destained. Right? Here's the destain intersection. And then finally, they go past a laser on the instrument and are detected. And they actually aren't directly stained, they're indirectly stained. So what we have is a molecule that essentially binds into a cell of RNA, and when it does, a conformational change occurs that causes an increase in its fluorescent signature. So we don't even have to find anything that specifically binds. It simply is a function of the shape of the molecule that we're analyzing. So as it passes by the detector, we get these very distinctive electropherograms. And this is, let's say, eukaryotic RNA. We have our 18 and 28S ribosomal RNA bands. Now, an intact RNA versus a partially degraded RNA, where there's a lot of this sort of baseline increases, it's very important that we know that that's the case. And then, of course, we have here what it looks like on the virtual gel. All right, so this is an important step upstream of your microarray or real-time analysis. Okay, workflow stuff. So from a workflow perspective, you probably want to create dilutions of your samples and standards. The good news is that we can handle such a broad range of inputs, from as little as 1 copy up to 10 to the 8th or 10 to the 9th copies. So you don't have to agonize over how much RNA you're adding or how much cDNA. The standards, if you're ever going to do standard curves, have to match the samples as closely as possible in every respect, including complexity, pH, salt concentration, all the things that matter. The samples themselves need to be quality controlled. We've already talked about that quite a bit. Then you set the plate up. You prepare all samples in replicates. Some people think that duplicates is enough. I'm a big fan of triplicates. Obviously, it's hard to decide which one's an outlier if you only have 2 values.
Mark, does the MIQE guidelines say duplicates, triplicates, which one is?
They have a discussion of the minimum effect size,
Okay.
right? But I think you would find them very pro-triplicate, or even quadruplicate. Of course, everything to them is a matter of, that's the focus of that research, is the real time. A lot of people, they just sort of want to confirm something else. It's not as much of a focus. Reaction volumes. Some instruments are now being designed, like the CFX, designed for 10 microliter reaction volumes. So that really is going to be an instrument-specific issue, what reaction volume you use. Primers, dyes, magnesium, everything can be optimized. But I'm going to try to talk you into just doing the simplest possible optimization. All right, this is my most controversial slide. And it's something that I think we ought, as a field, to talk about. And that is, how do we prepare replicates? It seems like a very trivial thing, but there's a lot of variability in how people are actually preparing their replicate reactions. So we always start with a master mix. And the master mix contains the super mix, which is almost always at 2x. It contains the primers, forward and reverse, water, and then whatever the detection chemistry is will usually go in the master mix as well. Okay? And that creates enough, let's do the math here, so 15, 17, 19, creates our 19 microliters times reactions. Okay? That's creating everything that's generic about the assay. And if we have 5 assays on a plate, we'll have 5 master mixes. But then you have a choice. Do you take your final reaction well and put your master mix in it and put the sample on top? Or vice versa, it doesn't really matter for me. Or do you have a separate tube where you prepare enough for 3 reactions for the replicate? Put it all together, right? Pre-pool the sample. And then from that tube, we aliquot to 3 separate tubes that are going to be run on the final plate.
I like that way.
Yeah, so do I now. My boss would never have let me do this. He would have said that's not a true technical replicate, right? Because the samples, right up to the point where you split them out, were all pulled together. All right? The upshot of this is that whatever was in here is chemically homogeneous. So when I sub-aliquot it, I'm not introducing any variability in my pipetting technique. So it eliminates the contribution of pipetting to variability. Right? On the negative side, some people don't feel it's a true technical replicate. You need to make sure you have no template controls and RT minus controls as well. And again, that's a requirement of MIQE now. So the no template control is where we use water instead of DNA. And the RT minus control is the cDNA synthesis reaction that we have not added any enzyme to. As you scale down, and again, many instruments are designed to be compatible with 10 and 5 microliter reaction volumes. You need to make sure that you keep the relative contribution of each component in the reaction as large as possible. You never want to pipette less than 5 microliters if you can avoid it, right? Because there's a fixed pipetting inaccuracy. And we want to try to avoid magnifying that by pipetting half a microliter of anything. Some of you might still be using old-fashioned conventional PCR protocols for your real-time, simply because you went straight from a conventional protocol to the same protocol on a real-time instrument. You might also be using very old hardware that required a long time to equilibrate. Newer instruments require much shorter reaction conditions. And besides saving you time, it can actually improve the quality of your data. Remember that in the reaction mix are triphosphate nucleotides, and those are actually quite heat labile. So at 95 degrees, we're going to be losing a lot of our DNTPs if we cook them for 20 minutes, or if we have repeated hold at 95 degrees for a minute. So we want to try to minimize the amount of time we spend at high temperatures. Now the protocol I've got up on the screen here is a typical older fashioned, let's say second generation protocol. It would be 3 minutes at 95, 10 seconds at 95 would start your PCR cycle, 55 for 30 seconds, and I'm making it hard to keep.
They is keeping me on my toes.
If you're using an ABI supermix, this would need to be 10 minutes. All right, they have a different kind of hot start. If you're using the newest generation of reagent, this can be cut down to 30 seconds, and I'm going to be talking about our latest generation's reagents. This can be cut down to 1 second, and this step can be cut down to, I've had good luck at 10 seconds, but it is possible to get it optimized down to 5 seconds as well. All right, and that's with really no major departures from what you're already doing. It's just a matter of putting different juice in the tubes. Right? And the reason why we don't need long periods of time is because your DNA is completely melted by the time you've hit 95 degrees. It's probably melting at around 88, 87 degrees. So by the time we've gone to 95, it's already completely melted. The same thing here. We've always produced such a huge excess of time, largely because the blocks themselves took a long time to equilibrate. So newer generation systems require a lot less time. And then lastly, we would tack on our melt curve if we're using cybergreen. If you're not, it wouldn't be appropriate. Oh no. I had the slides in here twice. So next section is analysis and quantification. And this, of course, is the meat of the presentation. There are two types of quantification. One is almost non-existent these days, and that is absolute quantification. The only people I still see doing on a routine basis standard curve-based ELISA-type absolute quantification are virologists, people looking at viral loads. Other than that, most people have switched to a relative expression or quantification. So we're only comparing sample A to sample B. We can do it directly or we can do it indirectly. I think we're going to mostly focus on direct comparisons of samples. So absolute quantification, there's one and only one way it can be done. It's the same way we've done ELISAs for many, many years, and that is we create a standard curve, and that standard curve is on every plate. Based on the standard curve model we create, we take an observed CT value for an unknown. We determine where it intercepts. We drop down and we say there are that many copies present in that sample. There's no ambiguity here. Now it's still somewhat relative because we're comparing it to a standard curve model. So it's relative to where it falls on that model. So in this case, we would say we have 10 to the 5th copies in that well. Okay? That's it. That's the only way it can be done. Relative quantification is a bit more complicated. There's been a lot more development in how we can do these kinds of comparisons. This was the original, the simple delta CT, or the relative abundance calculation. It was the first relative method to use direct comparisons. And it's just like the example I gave in the very first slide, or second slide, which says that if I have a treated sample and an untreated sample, and GOI here stands for gene of interest, and the difference between them at the CT is a delta of 3 cycles, then that's 3 rounds of doubling. 2, 4, 8. There's an 8-fold difference. Now, because I know that there's an inverse linear relationship, remember our standard curve model that had a slope down and to the right, that the smaller the CT value, the higher the abundance of the sample. So in that case, I know that the treated sample, which has the smaller CT value, is the higher abundance. So I could say that the treatment here caused an 8-fold increase in the gene of interest. It would be great if this worked. There's just one problem. And that is that we can't be sure that I put exactly 10 femtograms of cDNA in this one and exactly 10 femtograms of cDNA in this one and got the same number of relative copies. I also am not normalizing for any of the other effects, like the relative efficiency of the reverse transcription or cDNA synthesis. I can't be sure that I've accounted for all the off-target effects. So in order to normalize for all those load amounts, all those relative load differences, I'm going to do what we've done in Western blots going back several decades. And that is, we're going to strip and reprobe for beta-actin, or in this case, we're going to do a simultaneous assay for a housekeeping gene. And the key papers here, actually the first one to propose this was Mikkel-Thoffel. And I never realized the order that these things came out. And he came up with an equation that most of you will not recognize. This is much more common in Europe than it is in the US. For this to be part of the publication. And it looks very unfamiliar. But if you plug in 100% efficiency here and 100% efficiency here, you come up with 2 to the delta CP. If the efficiency is not 100% for both assays, then it doesn't reduce to that. This looks like that. And so this is the Libak and Schmidtgen paper, 2001, that first proposed the delta-delta CT method, or 2 to the minus delta-delta CT. And that's the equation that most people are using today. But again, the Thoffel method is simply the generic of the Libak and Schmidtgen method. The Libak and Schmidtgen method assumes, very importantly, assumes a base of 2, which means a true doubling in every cycle. And I've already shown you some cases where that was not actually true. So there's an assumption here. This equation only works in the special case that your efficiencies are indistinguishable from 100% efficiency. Okay? Then lastly, we have the Van de sample, or we might call this the Cubase paper. And this paper is definitely one you should get your hands on. It's PubMed Central available, so it's free full text. And that's Genome Biology 2002. 2 very key things came out in this paper. One was the proposal of how to use multiple reference genes. We already alluded to that we might get better normalization if we had several reference genes to use. But he also came up with an entire field of math for calculating the stability of a housekeeping gene. So instead of choosing it arbitrarily, we can base it on empirical observation. I'm going to choose beta-actin because it's the most stable gene in my conditions, instead of I'm going to choose beta-actin because everyone else is using beta-actin. So here's how we might do this sort of on paper. We've got a CT or CQ value for the gene of interest, and we've got a CQ value for the housekeeping gene, and we take a simple delta of those 2 values. if we are doing the von de Sample method, we take the geometric mean. There's a generic thing for it, but I just want to spell it out. The geometric mean of all the housekeeping genes in the study. Then we take the delta CT, which is simply for each sample, we take the delta CT from here, and we subtract the calibrator sample from that amount. So that gives us our delta CT. We take 2 to the minus delta CT, and that gives us our relative fold difference. All right, there are a number of software programs that do this for you. So the FAFL equation, instead of assuming 2, and that's not the actual equation, I just showed you the actual equation, actually substitutes in the actual efficiency. So it's always more accurate unless the efficiency is actually 100%. And then, of course, von de Sample is the geometric mean of reference genes. This is actually a figure from that 2002 von de Sample paper. And I think one of the most important ones for me. And this is 7 different treatments, A through G. And we're testing them for beta-actin and HMBS and HPRT and TATA binding protein and ubiquitin. So a variety of different housekeeping genes that are purported to be very stable. And what you can see here is that in an unnormalized comparison, these are the fold changes, that the difference between, let's say, A and B here for beta-actin, or I guess between G and B, is probably a 7-fold change. That's a heck of a big change. That's biologically interesting. But in fact, what we're doing here is we've got very poor normalization, because this gene is probably responding to something that's different between these 2 different conditions. And that makes it unusable in this case. And you can see that any one of these genes is going to have the same degree of variation from sample to sample. And that's unacceptable if we're going to use this to normalize our data. So there is no such thing as an invariant housekeeping gene. So if you want, you can kind of imagine if we took an average for the expression of these 5 genes, we might get something with a bit of variation to it. It would be a lot better than choosing any single gene from that panel. So the aggregate geometric mean of these values gives us better stability than any single value on that representative figure. All right, so multiple reference genes can make a big difference in the degree of changes that you can detect. No one wants to do it because, of course, it's more expensive and more work and more plates and less on each plate. But it does give better stability. So, I'm sorry, assay design, troubleshooting, and optimization. I'm going to go through this as quickly as I can. How are we doing on time?
So we'll up to 2.
Okay.
So another 20.
So the shortcut, and I love this, and this is also the work of von de Sample. The shortcut here is to go to his database, which contains user-submitted assays. And that means that if someone's published a real-time assay for a particular gene, they can upload it to this site, fill out a small form that goes through how the assay was designed, how it performed, maybe a figure or a graph, and all of that information is uploaded to the database for other people to search and use. This is part of the scientific community filling a need that everyone has, and that is for new assays. So you should never have to design your own housekeeping gene assay. They're all in here. There are over 9,000 assays. The search engine's not all that great. They're making improvements to it now. But this is a free database of publicly available information about assays. If you have to design your own, it is actually quite a lot of work to go through and do very meticulous design. And for some people, it's a bit of a barrier to entering real-time PCR. So we start with the bioinformatics, and there I really can't help you. You have to find unique primer sites. And in some cases, the database is not that clean. You may find a lot of sequences that match up with your primers that may be part of multiple sites. And that's when you get graphs or results like this, where you have all these full-length matches. And those are all probably, this one may be an EST clone, and this one may be a contig, and that one may be an image clone, all these different sources for the same sequence. Once you've got your unique primer sequences, you can test for secondary structure in your product. But you want to design your assays to be small. So we're usually looking for an assay between 50 and 150 bases in length. And that, of course, is very tiny when compared to conventional PCR, where we might be amplifying anywhere from 300 bases to several kb. Right? So we're always looking for very short amplicons. And that's additionally why we don't need these long holds. You don't need 1 minute at 72 degrees. You don't even need 72 degrees at all. Okay? Watch out for primer dimers. You need to make sure that you've used software to check for the presence of interactions between your primers. When they form, they tend to be very high efficiency, and they outcompete the specific products. And then lastly, we're going to have to test them in real-life conditions. And this is the part that gets expensive. So I want to talk you through the fastest possible way to optimize a new primer pair. But before we do that, I've got a troubleshooting example that I want to use to kind of hammer home the idea of validating a new assay. This is actual data. So this is real-world troubleshooting example. And it's very typical for what people see when they first start out. So here you see some new primers that were actually commercially purchased for a particular cytokine. And they're looking for it to be very high sensitivity, so very low levels of input. And they've done a standard curve of plasmid from 10,000 copies to 16 copies by 5-fold dilutions. So you would expect here 10,000, 2,000, let me highlight. This would be 10,000. This is 2,000 here. And then 400, let's see, what would it be? 400, 80, 16, and then no template control. And you can see that these last 4 dilutions here are all compressed. They're all basically the same CT value, plus or minus a little bit. So we're calling that no resolution below 2,000 copies. We keep diluting it, but there's no further increase in the CT value we should expect. The efficiency is 153%. The R squared is 0.957. Can anyone tell me what the likely cause is here? What did we say greater than 110% almost always was?
Primer dimers.
Primer dimers. So what's a good way to test for primer dimers? The gel, that's one good one. What else? Where did we see them before?
A melt curve.
Melt curve, very good. So let's run a melt curve on this data. All right, we're going to break it down by concentration. Here's your 10,000 copy melt curve. It has a melt profile around, my line's a little off, but let's say around 89 degrees. 2,000 copies, right? Around 89 degrees. 400 copies, aha! We have something that melts at 78 and something that melts at 89. So there are 2 products in this tube. And then the no template control, where we didn't add any sample, has a melt profile around 77 degrees. So, or 78. So whatever is amplifying here in the no template is also amplifying in the 400 copies. But it's not amplifying in the 10,000 or the 2,000 copies. There might be a little bump here. I mean, you can kind of see it's starting to bump up. So that is almost certainly primer dimer. Remember we said the break point was about 80 degrees? So there's your 80-degree break point. That's a low temperature product. So this is almost certainly primer dimer, and that's further reinforced by our no template control, where we didn't add any sample, but we still got product. That means this most likely has nothing to do with the template that we've added, unless there was contamination. Right? So what we're looking at here is primer dimer. But it's interesting that the primer dimer isn't a problem in the 2,000 copy. And primer dimer isn't a problem at 10,000 copies. This is what I call the teenager effect. In my observation that a bored teenager is the most destructive force in the universe. Right? If they have nothing to do, no matter whether they're good or bad teenagers, they will eventually get into trouble. And if you leave them in a room with nothing else to do and lots of toys hanging around, they're going to engage in something you don't want them to engage in. And primers are the same way. If there's nothing for them to do here in 400 copies, there's not enough for them to do to occupy them, then they'll tend to form minor products or unpredictable products or interactions between primers, just like teenagers. All right, so in this case, this assay is not a bad assay. It's not broken, but it certainly is unusable below 2,000 copies. And if we need it to work at that range, then this assay would not be acceptable. And what would happen, you would validate this assay and say, oh, this is a great assay, if you ran it at 10,000 copies and saw a nice clean curve. But then as you start collecting your samples, you find that even your negative controls, your uninduced or your control population, vehicle treated only, are also getting CT values around 26. And you say, gosh, they're all expressing this cytokine even when I don't induce them. Right? So this is why we have to validate not a single point, but a full range of inputs. We need to know across what range is this assay usable. And we want to do that quickly. Now here is the same assay, but with a TaqMan probe. A lot of people get the idea that TaqMan probes fix primer dimer. That's not really true. All right, we've got the same primers, but with a specific cytokine-specific probe. The efficiency is now 71%. 10,000 copies, 2,000 copies, 480, 16, and no template control. They're all still crammed together. All right, or rather, they're spaced out, but almost at random. And the R squared is 0.721, so not really a very good model. Now why? We've got specific probes. They're not binding to the primer dimer, right? Haven't we fixed that? But in fact, the primer dimer is still forming. It was a problem with the primers. It has nothing to do with the probe. The probe isn't detecting the primer dimer, but it's still interfering with the PCR itself. So until you have fixed your PCR, adding an expensive probe is not going to solve the problem. All right, they're still competing for reactants between the product we wanted to make and the primer dimer we didn't want to make. And here's how the assay looks once it's optimized. We get a 91% efficiency and a 0.99 R squared. So how do you optimize an assay? Does anyone know what primers cost? Someone in this room I bet does.
$5.
$5. Okay, $5 per primer, $10 for a primer pair. So your reactions probably cost you anywhere from 25 to 50 cents, discounting technician labor, which of course is free, for as far as I know.
Grad students.
Yeah, grad students are free. So you can do the math on that. If you spend more than 20 wells optimizing versus reordering a new primer pair, that's probably a bad financial decision. So we want to try to minimize any optimization and validation, but we still want to do it. So the fastest way to do it is this two-plate optimization. So here's your plate one. It's a gradient PCR. If you have a gradient real-time instrument, you can do all of this on the gradient instrument. If you have a gradient conventional cycler, you can do it on the gradient conventional cycler and score it sort of afterwards. It's not quite as good. So you really want to use a gradient real-time system, like the ones that Bio-Rad makes. We want to run a gradient of temperatures. Sorry, I've gone too fast. We want to test a range of temperatures. I usually do a 5-degree range from 58 to 63. Sorry, 50. Oh, put this back down. Sorry, I have to put the eraser down. From 58 to 63. And that usually tests 2 degrees below and 3 degrees above 60, which is where most of my assays are designed for. We're going to check two things for the same reaction, for the same replicate, run at different temperatures. We're going to look for no non-specific products. That means a single high-temp melt peak. And the other thing we're going to look for is the earliest CT value. So no non-specific products means that we don't have any primer dimer, and we don't have any pseudogenes amplifying. The earliest CT value tells us that we're at the most sensitive temperature. So for all of the specific temperatures, which one gives us the earliest CT value for the same sample? That's the most sensitive specific assay. If you remember from MIQE, one of the conditions was evidence of optimization from gradients. So that means they actually would like to see you have done this. And what you'll find usually is that a range of temperatures, in this case, this whole range corresponds to this CT value. Now, as soon as you go up to 64 degrees, you get a shift, a delta CT, or CQ. I should start switching over. A delta CQ. And that shows that you're less sensitive for the same sample. And of course, by the time you get up to 67 degrees, or 66.9, we're way out here barely amplifying at all. Now that also tells us, and this is something you suggested earlier, that this is a TACMAN probe TM that would be acceptable with this primer pair, because we're barely amplifying or we're not amplifying at all. Right? So if we designed a TACMAN probe, we'd want it to be above 67 degrees for this primer pair. That was plate 1. This is plate 2. This is going to be, for most of you, the only standard curve you will ever run, but you run it for each new assay that you get. Every new primer pair that you order up, you need to run your gradient PCR, and then you need to run your standard curve. You take any old sample that you've got that's positive for the gene that you're testing it for, and you dilute it a couple of times. Now, the range of your standard curve is going to be the range of your assay. So you want it to be above and you want it to be below where you expect your samples to be. You're going to get a standard curve, and the R squared might be 0.8, at which point you'll have to start Xing points off your curve until you find the portion of it that has an R squared greater than 0.95. Right? So we need it to be greater than 0.95. That's my own standard. You can certainly argue more or less. And then based on the slope of this line, we're also going to be getting the efficiency. So once you've found the R squared acceptable region, look at the efficiency. If it's between 90 and 110%, you have a working assay that's validated and optimized in 2 plates. All right, that to me is the fastest possible way. Now, what if the efficiency is 80% in the acceptable range? You can choose to use that if you so choose. I mean, you're going to get a lot of pushback from reviewers. Or you can throw those primers in the trash and order up new ones. And we've seen a lot of people move to ordering 2 forward and 2 reverse primers for every new assay that they design. Right? The advantage of that is not having to wait for the oligo company to design or to synthesize and send out the next batch. Does that make sense? Last thing, we're not quite done in our optimization scheme, and that is our gel-based analysis. So this will confirm the specificity of your assay and that it's actually producing the correct size product. You can't get that from a melt curve. So at some point, you got to break out the agarose gel rig and pour your agarose gel and run the products. All right? But that really, you can do this in a morning and be running in the afternoon. So it's not a lot of work. You just basically need 1 sample and you need a lot of it. A lot of people have started creating pooled samples. So they harvest a couple of cell lines, pool all the RNA together, and make one giant cDNA. And they use that for positive controls, optimization, validation, and anytime they want to do troubleshooting, because they know that it's of good quality and there's more than enough of it to throw away. Last area, assay considerations. So a couple of things, if you haven't already started your real-time program, make sure you have a clean area, especially if you work with plasmids or virus. They tend to get on everything. So if you have an area that never gets exposed to post-PCR products or plasmid, and even if that's a tissue culture hood, tissue culture hoods tend to be a little bit more isolated. You get fewer people reaching inside for a Kim wipe or a paper towel or something. Screw cap tubes, as opposed to the 1.5 mil Eppendorf tube, what a lot of us do is flip this open with our thumb, and we end up coming into contact with the rim there. I'm just having fun drawing. Tend to come into contact with the lid itself, and that contaminates the ball of your thumb, which then contaminates your pipetter. 2 mil screw cap tubes and good tissue culture-like technique are the best way to avoid contamination. Once it starts, you end up throwing away a lot of stuff. Aerosol-resistant tips, calibrated micropipetters, always pipette large volumes, minimal pipetting into each reaction well. And then this is the one I added to this screen, which is clean with bleach, not ethanol. I'm not a big fan of bleach. I actually prefer Formula 409 tub and tile cleaner, spring scent, right, because it smells nice. The point is that 70% ethanol, which is so effective at neutralizing bacteria, is completely ineffective at neutralizing DNA. Remember that we can precipitate and purify DNA in 95% ethanol. So 70% ethanol is just going to make it stick harder and keep it nice and safe. So we want to use something that actually oxidizes and damages the DNA. Very important that you choose the right reagents upstream of your real-time PCR. This is one of the areas that we see a lot of people making obvious mistakes in. And there's a lot of discussion about this in MIQE as well. We recommend very strongly that you use MMLV, that's Maloney-Murine leukemia virus, reverse transcription, reverse transcriptase, the enzyme from that virus. And the reason why is it gives the best representation and yield, and especially representation. Now, AMV tends to give better long RT, but we don't need that. We're doing 50 to 150 base pair products. We don't need 4 KB transcripts. And the HIV tends to work well for something that's 80% GC or more. Very, very important. This is the one element that is the most common mistake people make. There's a particular commercial RT kit. It's called Superscript, and it's from Life Technologies. And it was designed for a time when cDNA libraries were the hot, cool new technology. And it was designed specifically to give full-length transcripts so that they could be copied into clones and create an a transcript library. Right? We don't have that same application need anymore. We now need short chunks, but we need them from the full transcript. We need them all over. We don't want to introduce any bias. So with MMLV, there is an RNase H knockout mutant. And that's what you're buying when you buy Superscript. If you buy from a company, well, it's actually ABI and Life Technologies are now together. The ABI produced reagent is a native MMLV, and the Bio-Rad reagent, which is called iScript, is also a native MMLV. I'll put iScript up there. That'll make Randy happy. The RNase H is the second activity, other than the RNA-dependent DNA polymerase. It degrades the RNA-DNA hybrids, and it binds to the template primer duplex. Right? If you don't have it, you're going to be making a lot of first-strand product, but the RNA is still going to be bound to that first-strand product. And that creates a lot of problems when you go to make the second-strand product. So unless you are adding RNase H as a separate step after reverse transcription, you're going to end up with problems. This is a paper that explored that specific problem, and we haven't seen a lot of papers on the topic since. I think we're working on that in-house. But this is June 2002 Biotechniques, and they're looking at 3 different, I believe these are different splice variants, NCX1, NCX2, and 3, for the same sample from the same brain region, with and without, this is without and this is with, RNase H treatment as a supplementary step at the end of the procedure. If you do not treat with RNase H, the NCX2 disappears. And that's because it's being bound up by the remaining RNA, and it's preventing its transfer to the second strand cDNA synthesis reaction. So it's very important that you have an RNase H step somewhere in there. If you're going to use Superscript, make sure you do the supplementary 20-minute incubation followed by an activation of RNase H, or use a wild type MMLV. You have a choice of either oligo-DT or random hexamer priming during your reverse transcription. I highly recommend random hexamers or a blend of oligo-DT and random hexamers. Oligo-DT will tend to give more full length because, of course, it biases towards the polyadenylated region. And remember that you can't use 18S, which is not a polyadenylated product. It will not amplify an oligo-DT. That also, of course, means it won't be transferred in terms of mass. Random hexamers give you lots of little pieces along your transcript. And that's okay for us because we're producing very, very small products. It would not be appropriate if we were constructing a cDNA library, but it's very appropriate for real-time PCR. And then, of course, gene-specific means that you produce something in sort of a one-shot deal. So here also is the effect of CT value, or rather the choice of primer on the CT value. For two different RNAs, this one is not much affected. There's about a one-cycle shift if you're using random primers versus oligo-DT. There's only about a one-cycle lag. But for other transcripts, it could be 2 or 3 cycles. And that, of course, is probably larger than the kind of effects most of us are going to be looking for. So it's very important that we're consistent, and I highly recommend the random hexamers or random oligomers. The other thing to keep in mind is the length of protocols. So superscript has a lot of steps, ours has a fairly short number of steps, and I believe ABI's is sort of intermediate between. So make sure you shop around, find the right RT, and that it's a native MMLV. And then the protocol itself can be quite long, 45 minutes versus 85 to 90. So again, shop around. And then I call this the cell phone slide. As I walk around undergrad institutions like LSU and kind of peer in the doors, you can see people talking on their cell phone and pipetting with the other hand. You can always hear the music playing in a good lab. Just want to be aware of the distractions that prevent you from pipetting with any kind of accuracy. It's been my experience that it's best to isolate yourself when you're preparing the reactions. We are exponentially amplifying your errors, okay? So skipping a well obviously ruins your replicate. Do your best to stay focused. Last section. We do have a new generation of reagents. So these are called SOFAST, and that is a play on the organism that we stole it from, which is Sulfolobus sulpitericus. It's a deep vein organism. And what we've taken is not the polymerase itself, but a very small little peptide or protein. And we've attached it to a polymerase, both to TAC and to a proofreading polymerase. And what it is it's essentially a sliding clamp for DNA. Once the polymerase becomes associated, it prevents it from falling back off without a lot more energy. I don't know, some of you know what processivity is, but it's how far we go with our polymerase before it eventually falls off the template. And it's really quite startling. The processivity of TAC is usually 10 to 20 bases. So to make a 200 base pair product, we need 10 separate binding and extension events. Let me draw a little picture here. We need the polymerase makes, this product falls off, binds again, makes another product. So we need lots of little events. The SOFAST modification means that a single polymerase, it increases the processivity by a factor of 10. So a single polymerase makes the entire thing. And that makes this a very, very fast enzyme, but also a very high efficiency enzyme. So we can get more product made in a shorter period of time with less enzyme. And because it's stuck, because we've wedged it onto the DNA, it's very hard to knock it off with impurities or inhibitors that might otherwise prevent amplification. So some of the things that this makes it capable of: 1 to 2 kb product sizes. So you're no longer limited to the 50 to 150 base pair product size. And that's going to radically change, and we're a little ahead of the curve here, radically change how people design their assays today. We get 5 to 10 times the signal intensity from the dyes that we're using. So we're using with Evergreen the next generation of Cybergreen, which is about 5 to 10 times brighter. We can do direct PCR on unpurified DNA. For example, we're currently engaged in genotyping transgenic mice. So they have a transgene in their mice, and they want to be able to genotype that without going through any purification. And so they take 5 millimeters of tail snip, put it in a tube, vortex it, take 5 microliters, and use that in a reaction. So we go straight from urine, blood, tissue, whatever, directly into the PCR reaction with no inhibition in between. And that shortens up the cycle of time required to do the research. It's very resistant to inhibitors. So if you find yourself with a lot of phenylchloroform, if you pop the tube open and you can get a little whiff of chloroform, this is much more resistant to that. You get much better uniformity between your samples. It is an ultra-fast enzyme, so that's less than 30 minutes for 40 cycles. Lower volume reactions, so this is an enzyme that works quite well on our 96-well instrument at 5 microliters. We do have data at 3, but that requires that you have a very steady pipetter hand. And then it's a lower cost per reaction. It's a premium product, but it's actually selling for less than all of our other enzymes. And so this is the new dye. This is Cyber Green at different concentrations. So we've run it from everything from 1/4 to 2 times the standard Cyber concentration. You can see here at 1x, that's our full curve. At 1.5, and then 2 is a flat line, because twice the Cyber Green concentration than we would normally use completely inhibits the polymerase. All right? And that limits some of the applications that we can do with Cyber Green. If we need a saturating level of dye, for example, for high-resolution melt curves, Cyber Green is incompatible with cycling. At the level we have to use it at, it's incompatible with cycling. Evergreen is less inhibitory, so we can use a much higher concentration. So what is 1x here would be here several, 10x. So we can use a lot less of it. We can use a lot more of it without less inhibition. So some conditions exist for whatever instrument you're used to using. We have the protocols already written. It is very fast, so you can do a standard curve, again, in 30 minutes. And that requires a 1-second annealing extension, which is more than capable of producing a 200 base pair product in that time. It's also very sensitive, so this is single cell PCR. You take a single cell from LCM, and you amplify it and place it on the standard curve. And you can actually quantify how much cDNA or DNA is present in a single cell. And it works just as well at a standard old-fashioned 3-step protocol as it works at the 1-second annealing extension step. So the results are essentially matched. And then this is my favorite. This is the 1 kb amplicon on a 384 well instrument in 5 microliters, where we've got 30-second, 20-second, 10-second, and 5-second extensions. So unbelievably, you can make a 1 kb product in 5 seconds with this enhanced processivity of the TAC. Now that's with plasmid DNA. With samples, it's always a little more complicated. Any questions? All right. That concludes my presentation.
Shreveport, do you have any questions? No. How about at Louisiana Tech? No questions?
We are using the genotyping too, and I believe we are not using this so fast polymerase. We are using the TAC, the polymerase. It's TAC. I believe we use TAC, and works fine for us. And we use Cyber Green. But do you think potentially using this so fast will help us increase the sensitivity in the genotyping?
Absolutely. What are you amplifying from?
We're amplifying APP gene from the Alzheimer's disease mice.
You have mice.
Yes.
So you got brain regions, I assume?
We're actually looking at the tail like you described.
Well, we take the tail.
Yes.
And do you do any purification, or you take that directly in?
Directly into it.
Yeah, I think you'll find it works even better with this enzyme.
Okay.
Yeah, the signals are quite a bit stronger, and you probably see more uniformity between samples. And that's been our experience. Isn't that nice that you can skip over all those intermediate steps, right, and be able to look at the DNA directly?
Yes.
I have a question.
Please.
You said something about oligo-DT and 18S. What was that actually?
It's a simple thing, but 18S ribosomal RNA is not polyadenylated. So if you prime your reverse transcription or cDNA synthesis using oligo-DT, you will not amplify the 18S. Some people are using that for housekeeping gene.
Right.
We had a group very recently that couldn't understand why their 18S assay wasn't working, and of course they're using random hexamers only.
So you think if we are using oligo-DT, we should not use 18S as housekeeping? That's what you mean to say?
Yes. If you're using oligo-DT, you will not amplify any 18S, or you shouldn't.
We should not, but we do all the time. I mean to say, I do housekeeping genes, separate genes like GAPDH also and beta-actin as well.
Mhm.
But we do get amplification of 18S. And like we get CT value very less, like sometimes 6, 7
Are you sure it isn't a blend of two
of oligo.
primers? Mhm.
No.
Okay, well it's not polyadenylated, so it doesn't have an oligo-DT region. I mean, it doesn't have a poly-A region. So you shouldn't be starting anything
Right, but
unless you're getting little fragments of RNA that are acting as primers.
I know actually. That's why I was kind of.
Yeah, definitely go back and review that. I'm not sure I could find a publication for you that specifically says, but of course it's not polyadenylated. Anything else?
Anybody else?